Alright everybody, welcome back to part 2 of Cellular Respiration from Mr. Calvin's biology class. If you remember from last time, we were talking about glycolysis, which is a process by which we split up glucose to make ATP. And the net equation that we learned, shown right below. This is our last slide from the last video. Glucose, you can see right here, is broken up into two pyruvates, also known as pyruvic acid. Also what happens is we get, let me switch colors here so you can see it a little bit more easily. You get ATP being spent but also being produced. So 2 go in but 4 come out, meaning that we still have 2 ATP more than when we started, which is great. That's what we're trying to do. Also what happens, switch colors again right here, is NAD plus becomes NADH. And that NADH is later on going to become more of that ATP, that good stuff we're trying to make in here, which the cells use for energy. The overall equation for all this, as you can see right there, says that 4 ATPs are made, 2 are consumed, with a net gain of 2. Now that's just glycolysis. Remember, glycolysis does not happen in the mitochondria. This all happens out in the cytoplasm of the cell. So here's our mitochondria. Ta-da! And this beautiful little guy is what helps us live our lives because he produces all the energy. Remember, he's the power plant of the cell. All the energy that we use, and all that they ask in return is a little bit of oxygen and that wonderful chemical made by plants, glucose. Looks like I ran out of room there, but you guys remember what glucose is. That's C6H12O6. So, if we look at this, we can see some things that look very familiar to us. The mitochondria has an inner membrane and an outer membrane, just like cells and just like the chloroplast. It's a very similar organelle. Now, it also has something pretty important here. It's colored blue. In cells, the liquid inside, where all the chemical reactions happen, that stores minerals and other raw materials, is called the cytoplasm. In chloroplasts, this liquid is called the stroma. Here, this wonderful liquid, which contains all the stuff, is called the matrix. So the matrix is that liquid inside the mitochondria. That's where a bunch of the different reactions happen. The folds in the mitochondria are known as cristae. Now the cristae are where the electron transport chain is located, which we'll talk about a little bit later. Today we're going to focus mainly on the Krebs cycle, which is also known as the citric acid cycle, or CAC. But mostly we're going to call it the Krebs cycle in here. It's named after a guy named Krebs, and it occurs in the matrix. Remember that gooey, liquidy center on the inside of a mitochondria. The pyruvate, which was generated before in glycolysis, remember it's also called pyruvic acid, goes from the cytoplasm, not the cytoplasm, goes from the matrix into the mitochondria and is converted into a chemical called acetyl-CoA. That same acetyl-CoA then goes through a whole bunch of different reactions, which we're not going to go into here because all we really care about is what it makes. So we've got down here a nice list for every 2 pyruvates. And remember, 1 glucose makes 2 pyruvates. So saying for every 2 pyruvates is the same thing as saying for every 1 glucose that we're talking about right here. For every 2 pyruvates, we are making 2 ATP, so there's some energy. 6 CO2 is released as a waste product. Plants take in the CO2, we put it out. 8 NADHs, and 2 of something called FADH2. We'll talk about that guy in just a few minutes. Whew, look at that picture. Now, fortunately, we don't have to know everything that goes on here. Our starting point is pyruvate. Remember, pyruvate is the chemical that we had at the beginning of this Krebs cycle. It was generated by the end of glycolysis. So we've got our pyruvate right here. And as it goes across the membrane, we're going into the mitochondria right here. So we've entered the mitochondria. Now, once we're in the mitochondria, here it is changing into acetyl-CoA. But really what we care about is the pyruvate goes into the mitochondria and enters the Krebs cycle. So let's get rid of that, get that out of the way. I'm going to switch colors, make it a little bit easier to see what I'm talking about. Let's use green. Green seems like it would stand out pretty well right here. So the pyruvate comes in and it enters the Krebs cycle, where it goes round and round and round and round and round. Once the Krebs cycle is done, we make our CO2. There's our waste. We've generated some ATP. And we've made a whole mess of these NADHs, which are later on going to get used to make even more ATP. But you don't have to have all this memorized. It's just another way to look at it. Now, once we've gone through all the Krebs cycle, what happens is something called oxidative phosphorylation. And the key phrase there is this phospho part. When you change ADP into ATP, looks like I can't erase right there. Hold on, let me try that again. That's also trying something new. Sometimes you look a little bit silly. When you change that ADP into the ATP, you're adding one more P. You go from diphosphate to triphosphate. So what we're talking about here is generating good old ATP again, the energy molecule. So this happens in the cristae, those folds inside the mitochondria. And it involves the electron transport chain, which also shows up back in photosynthesis. It's just a way to pass high energy electrons along to transport energy from one part of a process to another part. And just like we saw back in photosynthesis, ATP synthase. All you need to know is that these two are involved. Also, something called a cytochrome helps out a little bit. The cytochrome here is a special protein that helps to pass electrons around. It's part of the electron transport chain. There's that transport part. Transport means moving. So the cytochromes transport or pass along electrons throughout the cristae. And all of this takes oxygen. Bam, that's the important part. So all this takes oxygen. It's aerobic, because remember aerobic means needing oxygen. And the end result is water, good old H2O, and a stunning 32 ATP. That's a whole lot compared to just the 2 or 4 that we saw earlier. And that's why mitochondria are so great. This fantastic 32 ATP that we see right there is a huge amount of energy. And that's the same energy that we need to carry out all of our bodily functions. That's why we bother to have these little mitochondria, these little mooters living in our cells. We're feeding them all this oxygen and all this sugar from our food, because in the end, once we finish this whole process, going from the very beginning at glycolysis through all of oxidative phosphorylation, changing ADP into ATP, all of that, is that we get 32 ATPs, lots of energy that we can use to do whatever we want. Now, the electron transport chain is pretty complicated. We've got electrons that move along from complex to complex to complex, just like a bouncing ball in a karaoke video. We got hydrogens going out of here to make a hydrogen gradient. The hydrogens then push through the ATP synthase. What you really need to know is we have all these different complexes. Complex is just a fancy word for a pump, for a protein that's going across the bilayer. See the bilayer right here, just like we've seen before? So what happens is each of these pumps hydrogens into this intermembrane space. This hydrogen, we're pushing it out of here, which means that out here, in between the membranes, we have got a lot of hydrogen. A lot of it. It builds up and builds up, making more and more and more pressure, so to speak. More and more concentration of hydrogen. And then all of a sudden, the ATP synthase comes along, and the ATP synthase, let's see here, sorry about that, that's this guy over here. The ATP synthase works kind of like a water wheel or a pump, And we have all this pressure of hydrogen built up on this side over here, and it pushes through the ATP synthase to get back into the cell. And when it pushes through, the ATP synthase turns kind of like a water wheel. And for every turn, for every hydrogen that it lets through, and that is a hydrogen right there, for every hydrogen that gets through, change colors right here, every one of these, we also change an ADP into, that's right, our old friend, ATP. So the whole purpose is we pump all those hydrogens out of the cell or into the intermembrane space where they build up pressure and then they rush back in through the gateway provided by ATP synthase. And each time they go through is kind of like a turnstile in a subway. They go through, it turns around once, and one more ATP gets made. It's an ATP making machine. Here's our overall reaction. So just like in photosynthesis, the fact that we have an overall reaction means this is pretty important stuff. And if you look at it, this is almost the exact opposite of what we've seen before. On one side, we have carbon dioxide and water, and a little bit of energy. On the other side of this reaction, we've got glucose and oxygen. Now, because we're not plants, because we're living things, we take in glucose and oxygen. Here they are over here, C6H12O6 and O2. And we get rid of the stuff that we don't need. The 6CO2, carbon dioxide, is waste for us, and 6 H2Os. But that is the exact opposite of the photosynthesis equation. In photosynthesis, we used the CO2 and the H2O to make our glucose and our oxygen. Here we're doing the opposite. We're using glucose and oxygen, and we're getting CO2 and H2O as waste. Here, in the photosynthesis reaction, we also had sunlight going into this, and that was our energy. In cellular respiration, we also have energy, only instead of sunlight, it's ATP that we're getting out of it. So what we're doing is undoing the work that the chloroplast did. The plant spent all this energy taking sunlight and converting it into stored sugars. We want that energy, so we eat the plant, we take that sugar and we break it back apart. We reverse it to get our energy back out of it. Here's the big picture overall. We got step number 1, we got step number 2, and we got step number 3. Glycolysis, change glucose into pyruvic acid. And it happens out here in the cytoplasm, not inside our mitochondria over here. So it happens just out in the cell. And that makes a little bit of ATP. Just 2. Not bad, but not great either. Then we've got the Krebs cycle over here, where things go round and round and round we go. And pyruvic acid gets changed into CO2, and we make a ton of NADH. And the Krebs cycle also, when it's finished, generates 2 ATP. So far we have a net gain of 4 ATP, and that's not bad at all. And finally, all of those NADHs that we were making over here in the glycolysis and over here in the Krebs cycle move on, and they're involved in the electron transport chain. Now, when the electron transport chain is doing, remember, is it's pumping the hydrogens out of the mitochondria, and then the hydrogens push their way back in through the ATP synthase, and every single time a hydrogen comes back in, it makes a new ATP. That means that this third step, even though it's not as detailed for us, we don't learn as much about it, doesn't make 2, it doesn't even make 4. Remember, it makes 32 different ATP. So this is definitely the main step. This is the big daddy of the steps right here. Here's where all the ATP gets made. But we had to go through glycolysis and the Krebs cycle in order to have all the NADHs that we needed to run the electron transport chain. So we have 2 ATP down here. We got 2 more ATP over here. We got 32 ATP over here, giving us a total of, that's right, 36 ATP. This is an important number. You need to know this one. You need to know, pardon me, let's put that back where that's supposed to go. Okay, moving things around a little bit. Okay, we've got this number you need to know, and this number you need to know, and of course this number. But the big thing, the total number of ATPs made by one turn of the overall process of cellular respiration is 36. Same thing again, looked at in a different way. Here's our 2 ATP, which we get through the glycolysis, happens out in the cytoplasm. Here's 2 ATP, we get to the Krebs cycle, that's happening inside. And then we have the electron carriers, NADHs, carrying those electrons, carrying those hydrogens to the electron transport chain, and that's where we make our 32. So the electron transport chain is definitely the highest yield. That's where we make most of our ATP. And that's it. That's it for part 2 of this. When we come back, we're going to talk about anaerobic respiration and all the reasons why all the foods that you've always loved to eat are actually secretly disgusting. Stay tuned. And